أعزائي طلاب المرحلة الثانية، السلام عليكم ورحمة الله وبركاته. راح نبدي شرح توضيحي عن مختبر التيومر ماركرز. المختبر طبعا أرسلنا الباوربوينت على الكلاس روم، وإن شاء الله هذا التسجيل الصوتي أيضا راح يوصل على الكلاس روم. بداية التيومر ماركرز مثل ما تكلمنا عنها بالمحاضرة هي عبارة عن molecules of two types and either are secreted by the tumor or associated with the tumor, what's called the tumor associated antigen, or they are tumor specific antigen. And this classification is represented by the lecture clearly. Here we will talk about different points, practical points regarding the tumor markers. The first point is the diagnostic potential of these markers, for how much we can use the tumor marker to diagnose tumors, and also how we can measure these markers, by which assay, and also we'll take a single most important and most common assay for measuring the tumor markers or testing the tumor markers, which is the ELISA technique. So this is about the content of the lab. First, we will talk about the diagnostic potential of the tumor marker. We have certain routine markers specific for certain tumors, and all are ordered routinely in order to diagnose tumor. But here we will see that these markers are not diagnostic and are not pathognomonic for the presence of the tumor. That's to say, when we find a positive tumor marker or an elevated tumor marker level in a patient, this alone does not mean that there is tumor. We should have a previous clinical suspicion and certain radiological and imaging finding, and also other biochemical laboratory criteria for the diagnosis of the tumor. So the tumor marker is one of the biochemical criteria for the diagnosis, and it is not the only criteria for the diagnosis. This should be kept in mind, and this is very important, and it is necessary to avoid the overdiagnosis of the tumor markers. So as we say, the interpretation of tumor marker results need excellent clinical judgment and excellent decision making to avoid the overdiagnosis. We should not rely only on the tumor marker to diagnose the tumor, and this is a very important point to be kept in mind. The other point in the diagnostic potential of tumor marker is that not all tumor markers are important only in diagnosis. Some are important in the follow-up or prognosis or even staging of the disease of the tumor, such as human chorionic gonadotrophin. This hormone is elevated in case of benign tumor of the placenta and also in the malignant tumor of placenta, choriocarcinoma. And the level of HCG might determine the progression of benign H-mole into malignant choriocarcinoma. Also, other tumor markers are regarded as therapeutic targets. So we have diagnostic application, prognostic use, and also therapeutic targets of some markers, such as here we have the most important and most common therapeutic target, which is the HER2/neu. HER2/neu receptor present in some kind of breast carcinoma make this kind of tumor vulnerable to certain therapy targeting this HER2 receptor, which is a growth factor receptor, and the therapy is called Herceptin, and it is routinely used to treat some cases of positive HER2 breast carcinoma. The other slide show a list of common markers in routine use to aid in diagnosis of certain tumors, such as CA15-3 and BR7029 for breast carcinoma, and also carcinoma embryonic antigen CA19-9 for colorectal cancer. And we have the same markers for gastric tumor, and also prostate specific antigen for prostate cancer, CA125 for ovarian cancer, and some other important markers listed here in this table. This is very important to have an idea about these markers and the application of these markers in the routine clinical practice as aid in the diagnosis. Please keep in mind that these markers are just reinforce the diagnosis. We should have prior clinical suspicion and also radiological and biochemical investigation. Well, now we proceed to other point in the lab, which is how to measure these tumor markers. Many of the tumor markers are measured by ELISA test, enzyme-linked immunosorbent assay test. This is an immunoassay. We will take it in detail in the end of the lab. So many tumor markers, most of tumor markers are measured by ELISA. Genotypic tumor markers. This means that translocation tumor markers are detected by molecular tests such as PCR, polymerase chain reaction, and karyotyping, and DNA sequencing, and even by flow cytometry. And some other markers are detected by immunohistochemistry. This is immunohistochemistry. It is immunohistochemical test, mixing the immune principle with the staining technique of the biopsy tissue, and this is widely used in the pathological aspects, regarded as pathological mean of diagnosis. So we have ELISA, PCR, karyotyping, DNA sequencing, flow cytometry, immunohistochemistry, etc. And some tumor markers are hormones, as we mentioned in the lecture, and these hormones are measured by variable biochemical procedures. Here we have example of the chromosomal translocation. Tumor markers represented by chromosomal translocation. We have two examples here. The first example is translocation between chromosome 9 and 14. Chromosome 9 and 22. And this translocation have characteristic feature of the chronic myelogenous leukemia. CML diagnosis is widely dependent on the presence of this translocation, and this translocation is termed Philadelphia chromosome. The translocated chromosome number 22 with missing long arm is called Philadelphia chromosome. The long arm of the chromosome 22 translocated into the long arm of chromosome 9, as we see in the figure. So Philadelphia chromosome is certain specific genotypic disorder or defect, is characteristic of chronic myelogenous leukemia. The other chromosomal translocation genetic defect occur in Burkitt lymphoma, characterized by the translocation between chromosome 8 and 14, and this translocation is termed c-MYC gene, c-MYC gene, and this c-MYC gene is characteristic for the presence of Burkitt lymphoma. Lastly, in the lab we have few words about the ELISA technique. You will see in the last slide some links, video links, in this video to show how we proceed the ELISA and what is the ELISA principle and technique. As you know, ELISA is referred to enzyme immunosorbent assay, and this is a quantitative immune test. That's to say, it measures the concentration or amount of the marker, not only the mere presence of the marker. So it is a quantitative test. The main principle, the broad principle is linking the antigen-antibody binding. So we make use of the specific binding between certain antigen and antibody, and we will show this binding by adding a further step, which is enzyme substrate reaction, forming a color, a substrate called the chromogen reacted to form a certain color in the presence of specific enzyme, and this enzyme will bind to the antigen and antibody, as we show in the figure and the video. So we have antigen antibody specific binding, and there is an enzyme substrate bound to this antigen antibody complex, and finally we will have a color, and this color, or termed optical density, that's to say the intensity of the color, this is measured by spectrophotometry during the ELISA test, and the amount or the number of OD is longitudinal or correspondence with the, or directly corresponding with the concentration of the marker to be tested, whether the antigen or the antibody. So ELISA technique here will try to detect antigen or antibody in the sample by using the color density of the reaction. Finally, the OD correlates with the amount of antigen or antibody in the test, and we will have the apparatus make certain curve to show this concentration, whether it is negative or positive, and the amount of this marker to be tested. Here we will show the ELISA. In this slide, ELISA can identify the presence of a particular antigen in a sample, and so determine if a person is infected with a particular disease, or a person having certain antigen or having certain antibody, etc. Example, HIV tests, and this ELISA assay is carried out in 96 well plates. This plate shown in this figure have 96 places for putting a sample, 96 small tubes. So these are termed wells, 96 wells plate, and sometimes it's called micro-titer plate. This plate with 96 sample, sometimes called micro-titer plate, or even it is called ELISA plate. So the ELISA reaction or tests are carried on in these wells, and each well represents a separate sample. So we have run about 90 samples in every ELISA plate or every micro-titer plate. So what is exactly going on in each well? There is several steps of addition and incubation washing steps. Between every step and the other there is washing, and frequent washing is very important. We have a video in the last slide show how these addition and steps are going on. Here in this figure showing the ELISA types, we have about 4 to 5 types, and these are the main 4 types of ELISA test, starting from the left by the direct ELISA and indirect ELISA, sandwich ELISA, and competitive ELISA. The direct ELISA search about the antibody, so the wall and the ELISA plate is coated by the antigen, and the sample might contain the antibody. As you know, as you see in the figure, the first from the left, which is the direct ELISA, we have the antigen who is lowermost, and then the antibody. This comes from the patient sample. After that, we have the enzyme and substrate on the top. While for the indirect ELISA, the same idea, we will have a bound antigen to the wall, and then we have the sample contain antibody in a question, and we have secondary antibody bound to the enzyme and substrate. In sandwich ELISA, the third type, we have the wall coated with antibody specific for the antigen in a question, which is occur or found in the patient sample, and then we have a bound specific antibody and another secondary antibody, and then on the top we have the enzyme and substrate, which give the color. For the competitive ELISA, we have 2 antigens, one bound to the wall and one is edited as inhibitory antigen, and the amount of the color will indirectly correlate to the concentration of the antibody in a question. Going back to the first type, which is the direct ELISA, if we talk about the steps of the ELISA, we have the ELISA plate containing 96 walls. In the walls of the sample, we have already bound antigen. We add the sample of the patient, which might contain the antibody in question. Antibody, if it is present, will bound to the antigen, and then we will wash unbound material, and then add the enzyme substrate concentration. Enzyme substrate will form a color according to the amount of antibody present in the wall. So finally we'll have a color, and this color is measured by optic density and correlate the concentration of the antibody in the sample. And the other types of ELISA, we can see the differences in the steps according to the presence of the bound antibody or antigen to the wall, and according to the what we search about, which is either the antibody in case of direct and indirect ELISA, or antigen in case of sandwich ELISA and competitive ELISA. Competitive ELISA search about the antibody also. So only sandwich ELISA search about antigen. So the next slide talk about the ELISA procedure. As we mentioned, the first step, we use the ELISA plate, or this 96 wall plate or microtiter plate. This plate contains 96 wall for the sample and the standards. Standards here are put to mount certain calibration to the curve of the concentration. Standards contain certain known concentration of the material in question, and these standards will make use to draw the standard curve of ELISA. This is done automatically by the machine. We just put some 5 or 6 wells as standards to draw the curve. Okay, so the well contains samples, and the standards are put in the wells, and several steps of addition of antigen or antibody or enzyme, substrate, and etc. And there is a period of incubation, and there is steps of washing between every subsequent step, and the video explain more these steps. By the way, every ELISA kit have certain procedure, and it is explained by the ELISA catalog of the kit, and the researcher or the laboratory worker will follow the instruction fixed in the catalog of each kit. Now for the result reading of the ELISA. After completing the run of ELISA, we will have OD numbers. Every well of the 90 wells will have certain OD number, and this OD number represents certain concentration of the material in question, whether the antigen or antibody, and this is drawn by the or represented by the ELISA curve. Here we have in certain details about the ELISA results reading and interpretation, and some kits have certain cut-off idea for the negative and positive measures according to the reference data. You can watch the videos represented by the links in the last slide, and you will see the procedure or protocol of ELISA in details and in a simple manner, and we are ready for any question you have, and thank you very much.